So the Krebs cycle, it's a cycle, yeah, but I like to break it down, or at least the book breaks it down into two stages. One of them being the decarboxylation and then the oxidation of the two carbon atoms, producing high energy electron carrier. First one of these enzymes that I wanted to mention is, chemically speaking, at the beginning of the cycle, quote unquote, citrate synthase. Broken each and every one of these enzymes down into their mechanisms and then sites of regulation, if any. But first, let's talk about the mechanism. So the mechanism of citrate synthase has an ordered binding mechanism. So what ordered binding means is where one substrate comes in and it binds, and that causes a conformational change that enables the other to bind, and then we have the reaction that's catalyzed. And the way that we do this is to avoid useless side reactions that would make our enzymes basically redundant. So first we have binding is oxaloacetate, and then second to bind is going to be the acetyl-CoA. And the reason that we do this is to avoid useless side reactions. And so what we would usually do is acetyl-CoA is really readily hydrolyzed. Like, that's something that's very likely to happen. However, we're not going to do this. We only want to hydrolyze the intermediate, which is citryl-CoA. We need to make sure that we're not doing that. So first oxaloacetate binds. This is going to cause a conformational change in the enzyme that is going to enable the second binding, which in this context is acetyl-CoA. Binding. Hopefully I'm not spelling that right. Anyways, in this second binding then results in the hydrolysis of our reaction here. We want to hydrolyze citryl-CoA as the reaction intermediate, and we're not going to hydrolyze the acetyl-CoA, as it would make it totally redundant. Anyways, as for regulations, there really isn't any. The next enzyme that I wanted to mention would be aconitase. And, like citrate synthase, aconitase has really minimal regulation. But the mechanism of it's pretty interesting. The mechanism is first to have dehydration, and then second we're going to have hydration happening. So what we're doing in this is so that we can orient the OH group, the hydroxyl group, for oxidative decarboxylation. And that's one of the main running themes of the first stage. And I'm just going to draw really briefly the reaction intermediate for it, which is cis-aconitate. The next enzyme is Isocitrate Dehydrogenase. As for the mechanism, we just oriented the hydroxyl group for the oxidative decarboxylation. So obviously, the isocitrate dehydrogenase, the next step, is to do the actual oxidative decarboxylation. Aside from the actual products that are pertinent in terms of the cycle, this is going to give us CO2 and NADH, so we've made yet another high-energy electron carrier. As far as regulation goes, this is one of the secondary regulation sites that we have here, and the way that we do this is we have certain activators and inhibitors, as you could imagine, that mostly work through means of a negative feedback loop, which is, since that's nothing really new. Activators, we have ADP, and then for inhibitors, we have NADH and ATP. One thing that I wanted to make about isocitrate dehydrogenase is that obviously if it has effector molecules acting on it, it is an allosteric enzyme. The last enzyme that I wanted to mention is alpha-ketoglutarate dehydrogenase complex. And this guy is kind of, I think of it like an allosteric enzyme. Obviously, if it's a complex, it's more than one enzyme, but it's regulated very much in the same way. And the inhibitors, as you could imagine, is resulting from that of a negative feedback loop. The activators, I mean, that's kind of a given what the activators are. All this is kind of revolving around negative feedback and the energy charge. So none of this is really conceptually new material. But for the inhibitors, we have succinyl-CoA, NADH, and ATP. And as for the mechanism, I know I probably is going to look crowded, but I promise you I can probably fit this. Is the same mechanism as what we saw with isocitrate dehydrogenase. It is an oxidative decarboxylation. Sometimes this video software will zoom in and I've had stuff missing, so hopefully you can still see that. But anyways, for the oxidative decarboxylation, this is going to give us, as you can imagine, CO2, an energy molecule of NADH, and what's unique to this though is that we have that the high energy thioester linkage. It's not really so much with stage 1 enzymes as I'm talking about the Krebs cycle as a whole. So you mentioned, the enzymes that we can have that sort of regulate the rate of the Krebs cycle, but the primary way that we regulate the rate of the Krebs cycle is through the pyruvate dehydrogenase complex, and basically anything that's going to ultimately affect the concentration of acetyl-CoA. Now there's lots of things that can give us acetyl-CoA, one of those being the pyruvate dehydrogenase complex, the PDH complex as I like to think about it. Basically anything that results in the production of pyruvate, right? So amino acids can play a role in this, glycerols, things like that. Just anything that they ultimately can give us pyruvate, which we can use to convert to acetyl-CoA. But that's the main regulator of the Krebs cycle is really the concentration of the beginning. The other enzymes, isocitrate dehydrogenase, alpha-ketoglutarate dehydrogenase, those are kind of secondary regulators.